phenomenon about viruses, and that's defective viruses. And I don't know if you've heard about them, but you'll know more than you may want to know about. So we're all familiar with multiple types of viruses, and here's a map from a few years ago, still relevant today, that shows a lot of these emerging and re-emerging viruses. And what's actually interesting about this map is that the majority of emerging and re-emerging viruses are actually RNA viruses. Now we know RNA viruses have a higher mutation rate than DNA viruses. So here's a typical representation of sort of the mutation rate you see on the x-axis, and you see how the DNA viruses have a much lower mutation rate than RNA viruses, and you see flu placed right here. Now, the reason RNA viruses have a high mutation rate is because they have an RNA-dependent RNA polymerase that doesn't have a proofreading function. And you'll see why that's important when we talk about defective viruses. Now, in the case of influenza, when you talk about transmission, so aerosolized transmission, in fact, these defective viruses probably contribute to about 90% of the particles that are transmitted. That means that for an infection to actually take into a recipient, it doesn't require that many functional molecules or functional viruses themselves. Now, what's interesting is that the defective viruses can also contribute to the infection, and you'll see how they have a role to play. They're not just junk, okay? Now, if you know nothing about flu, here's a pretty picture from the internet on flu, and then the genome is actually 8 segments, so it's a bit analogous to chromosomes. They're different sizes, and the largest ones, the 3 largest segments, code for the polymerase. And the way the segments are structured, they're actually this negative strand RNA that is intertwined with this nuclear protein, and they're actually folded into this structure. And the little yellow boxes you see on the top actually represent conserved ends of the segments, and they're very, very well conserved. They're conserved across all flu subtypes. So when I talk about H5N1, H3N2, H1N1, it's the same ends, and they actually base pair match on the end. And then you have this twisty structure. And on the top, you have this polymerase complex. And the polymerase then does the transcription through the whole structure. Now, when I talk about errors or variants or defective in the viruses, there are different types of diversity that the virus actually does. And here's a crude representation of 3 types of variants that the virus can make. In fact, if I take you to the last one, the reassorted genomes, flu, because of the segments, if you have 2 flu particles, flu viruses that infect the same cell, the progeny virus can be a reassortant of the different segments. And so that's not an error, it's really part of the life cycle of this virus to do diversity. But there's another way of doing diversity is by errors, and it takes you to the first one. So these mutations are when the RNA polymerase actually makes these errors that are not corrected. And that's also important to increase diversity of the population of the viruses. Now, there are other types of variants. There's what's called a semi-infectious particle. And that's where you can get a whole segment that is missing. So instead of having a genome with 8 segments, you can have just 7 segments. And then there are the defective viral genomes. And those between the mutation errors and the DVGs, the defective virus genomes, are due to these replication mistakes that are made. And so my focus today will really be on these DVGs, or defective virus genomes. So what does it really look like? You have basically this RNA-dependent RNA polymerase that will read along the molecule and then do some kind of skipping, and you'll have a shortened segment with a whole middle that is missing. That means the ends of the segments are conserved. And these segments, because the ends are conserved, can be packaged in the progeny. So you'll have virus progeny that have these much shortened segments. It's thought to be an error. And in fact, it was thought to be solely an artifact of cell culture. So if you do any type of virology, you'll know that when you culture a virus, you try to keep your infection process at a low MOI. That's a multiplicity of infection. So what does that mean? If you look at this graph, this was actually defective particles or defective viruses were identified back in the 70s by Alice Wong and David Baltimore. So it's been an old thing around for a long time. And what it shows here is they had made this model showing that you could have these functional viruses. So it's like these hairy things they represent here. And you'll have a low multiplicity of infection. In this case, one virus particle entering a cell. That virus will replicate, make multiple progeny, and that at the next replication cycle, you can have a high multiplicity of infection, meaning you have multiple virus particles that are infecting the next cell. And as the polymerase continues to transcribe, it can make mistakes and do this skipping that I was telling you about. And when it does this skipping, it can generate these much shorter genomes, but because they're co-infected with these functional genomes, they get replicated because they use the proteins from the functional virus, the functional polymerases. And then what happens is that the polymerase will preferentially replicate much shorter segments. It likes shorter segments versus longer segments. And so eventually what you have is a culture full of defective viruses, and it completely represses the amplification of functional viruses. And so we call that process a defective interfering process. So basically these DIs are defective viruses or DVGs that are interfering. Not all defective viruses are interfering, but all interfering viruses are defectives. So in our own experiments, if we take flu and we determine how well the virus grows, if we grow flu on its own, which would be the orange bar on top of this graph, it shows that it grows pretty well over a course of an infection. But if you add a DVG that is interfering, you can repress the growth of the virus. So DVGs can interfere with the production of the functional virus. So I mentioned that it was thought to be mostly a function of cell culture and an artifact in cell culture, but a few years ago, we actually noticed that it was present, all these DVGs were present in natural infections. So we see this in flu, we see this in West Nile virus, we see it in now multiple natural infections of RNA viruses. We have these defective viruses that exist. And here in this first study in 2011, we had looked here on the left, this is just a phylogenetic tree of flu collected from college students at UCSD. And what we noticed when we looked at the deep sequencing, we noticed that a lot of these individuals were carrying these defective segments for the polymerase. So you can see here, 2 of the polymerases have defective segments. And what we saw is that 2 of the college students who appeared to be roommates also shared some of these defectives. So that's what we're intrigued about. What are these defectives doing? Why are they present at such a high level? And does that have an effect on the infection per se? Now, we know very little on the diversity and the structures of the DVGs in influenza. So we did a pretty deep dive on that, and I'll just show you a few examples. And also the impact of the host on the evolution of the virus and on the presence of these DVGs is something we're very intrigued about. And then we'll also look at the host response to these DVGs. If they're interfering, they're triggering something in the host specifically. And that's what we've been looking at. So I'll just take you very briefly across these 3 points. Now the models we use, so we do natural infections, so we take samples from human infections, but we also do infections in ferrets. Now, why ferrets? Well, it turns out that ferrets are like little people. They have very similar respiratory tracts. They're social animals like we are. They sneeze on each other. They touch each other. They get similar fever. They recover from the flu in a very similar manner. And so they're a great model used for transmission. So when people are doing a lot of immunology studies on flu, they'll use mice because there are a lot of reagents available. But when you look at transmission and even at the evolution of the virus, ferrets are much better. So a typical setup of an experiment with ferrets is basically like this, where you would insert into the nose of the ferret, you would just squirt in some virus. In this case, it's the 2009 H1N1 pandemic strain, which is basically now during flu season is circulating and has been circulating since 2009. And then we basically take samples from nasal washes from these ferrets at multiple time points over the course of the infection. And we can also put these ferrets co-caged, so they're in cages together. They can transmit to each other, or they can be in separate cages and then they can aerosolize transmit. And I won't go into the transmission aspect of it. I'll just tell you about what happens in the original ferrets that are infected. Now, the process is pretty simple. We do the sequencing of all the segments, and we do here a typical mapping of the reads. It would look like this, just a profile of all the coverage of the reads for each of the segments. But when you have a population that has mostly defectives, what you see is something like that, that looks like what we call Batman ears. So the reads have missing internal segments, and that's what we observe in a lot of these natural infections and what we see in our ferrets. So basically the identification of the DVGs is very simple. It's based on split reads. And what we record to determine the type of DVG or the diversity of the DVG are the coordinates of the beginning of the gap and the end of the gap. And using these coordinates, we basically name our DVGs accordingly. And so using that very simple and straightforward method, we can look into the dynamics of the DVGs over the course of an infection. Are the deletions always in the same spot? Do you have hotspots of deletions? What kind of diversity do we see? Is there an effect of the size of the DVGs? A smaller segment has a different effect than a larger segment. And also the position of the deletions specifically on the segment. Now, to look at this, we've done multiple types of experiments. And one of the experiments we did a few years ago was actually to look at, we were mainly interested at the time, not even in DVGs, but simply how does the virus evolve differently in individuals that are pre-immunized with flu. So let's say you're a 40-year-old today, you've had multiple decades of exposure to different antigenic variants of flu. So you have antibodies to some of these antigenic variants. Now, if you're a baby, you haven't been exposed, so you're considered naive. Does the virus evolve differently in somebody that's had multiple exposure versus somebody that's never been exposed? Also, depending on your age, the first flu you were exposed to may basically sort of set up what you will respond to and how well you will respond to a further or later on strain. So in the design of this experiment, we had a bunch of naive ferrets that we infected, but we also had subgroups of ferrets that were pre-immunized with different decades of antigenic variants of H1N1. And we just wanted to look at the diversity of the virus, which I won't go into, but I'll go specifically into the DVGs because it turned out that that was more interesting than actually the variants that we could observe. So these ferrets were pre-exposed, they resolved their infection, and 12 weeks later, they were challenged with the actual California H1N1. And then we collect nasal washes at different time points, and again, do this mapping, do the split read alignment, and then determine the sizes of the DVGs. So if I look at the stock viruses themselves, This is a representation of, on the x-axis, the DVG size, so how big the DVGs are, with the frequency of these different sizes. And you can see that in the stock, we had a lot of DVGs and they were very small. And here I'm focusing only on one of the polymerases because it is the segment that gets deleted the most often. And so when we looked at our naive and at our pre-immunized ferrets, what we saw is that over the course of the infection, what you have is you're infected with this mix of a lot of small DVGs. There are probably a few longer DVGs, but early on, you have very large segments and very few of the DVGs, except if you've been pre-exposed. So in the pre-exposed, you seem to keep an accumulation of the smaller segments. And over the course of the infection, as you resolve the infection, you basically have smaller and smaller DVGs. Basically what we see in culture, where you would have higher multiplicity of infection and smaller DVGs. But that really is correlated with also how you resolve the infection. So when I talk about big DVGs and small, sorry, I'm losing my voice. So when I talk about small and big DVGs, this is what I mean. So a small DVG is where you would have a very large deletion, and a large DVGs where you have a small deletion. So this is what it looks like. So one of the questions is this a random process? Does it just happen constantly and you'll never find the same DVGs in individuals? Does the polymerase just skip all over? And so we decided to test this. If it was totally random, it would look like those gray spots where I would have the coordinate of the deletion start, what we call the gap start on the x-axis, and the coordinate of where the deletion ends, so the gap end. If it was random, you would have a start anywhere and an end anywhere, but in fact, that's not what we see. We see a very structured way of where it stops and where it starts and where it ends. And in fact, what we see is that at these deletion junctions, we have repeated nucleotides, so that what appears to happen is that the polymerase skips by recognizing something else further down on the segment. So if we go back to my structure in 3D of what the segments look like, what happens during the copy of the segments is that you have basically the nascent strand that grows as it reads down that side of the segment, and then eventually you have a full-length copy of that segment. So if I do my little cartoon here, what you have is that nascent strand that is going down, 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 and all of a sudden, we think what happens is that it skips because it recognizes another side. So in fact, what we see is that all the DVGs are very symmetric because it's recognizing something that is in geography on the structure very close. And so over the course of the infection, what we do see is that we have more abundant symmetric DVGs. So they become smaller, but they're very symmetric over the course of the infection. And so we started looking at all our data in human samples, in different infections, flu B, different lineages, different subtypes of flu A, and we always see this very symmetric representation. So there's a very clear structure to how these DVGs are generated. It's not random. Now, we know the structure and we're looking at a lot of the diversity of the DVGs, but what we're also intrigued by is how does that impact or how does the host modulate these DVGs? And does it make an effect on the diversity of these defective viruses? So we had another set of experiments where we had many ferrets where we could collect nasal washes, but also portions of the lung, the upper lung, the lower lung. So when you get infected with flu, normally it'll stay up in the nasal cavity, but you do have some viruses that can go lower down. In the case of these ferrets, because we squirted it into the nose, it goes down also into the lungs. And we thought you should have the same population up or down. It should be pretty similar. So when we look at nasal washes in the upper lung and the lower lung, and we look at just the number of reads that we find that have these deletions, we see that in nasal washes, it actually goes up in these ferrets. So over the time course, you get more of these DVGs, while the infection actually goes down. So this is a representation of functional viruses. So it would be plaque-forming units of viruses. So this is natural, what you would see in a natural infection, where you get a lot of flu and then little by little you lose it because you recover from the infection. But DVGs remain pretty high, at a high abundance in all the sites. And we see it's a higher abundance actually in the lungs than in the nasal washes. We also see that the DVG diversity is much higher in the nasal washes than it is in the lower lungs. And we see this with a higher diversity over the course of the infection in the nasal washes. But what was really interesting is that these ferrets, we know how sick they get over the course of the infection. So if I infected this room with flu, although even if you're healthy adults, no matter your age, 25% of you would probably have severe flu, 25% would be completely asymptomatic, although you have the infection, and the rest of you would have would have a little bit of disease, but it's moderate, what would be moderate. So you have the mild, moderate, and severe. Ferrets, it's exactly the same thing. And you can never know in advance. However, when we looked at the DVGs at day 1, and then we knew what happened to these ferrets at day 5 or 7 or 8, whether they had a severe disease, we saw that the mild group had early on a lot of these very short DVGs. So this is interesting because DVGs can be interfering. And a study a couple of years ago had seen that in flu patients, the severe cases were associated with people who had very few defective viruses in their infection. So more DVGs, you would have a milder infection. Fewer DVGs, you would have a more severe infection. So that brings back Nigel Dimmock, who is a scientist in the UK and incidentally the cousin of one of our colleagues at NYU, totally by chance. And he has been working for decades actually on DVGs. When nobody was working on it, this guy was working on these defective interfering. And he has been pushing for DVGs as antivirals, so completely different than what we have right now. A new way of controlling for virus infections. But what we need to do with these, understanding these defective interfering of these DVGs is to really know what's happening at the cell level. What is the response to these defective viruses? And that's what we looked at in a study with one of my students, Chang Wang, is a PhD student, and which it is a simple setup where she did a high multiplicity of infection because we wanted DVGs in this case. And she used 2 cell lines, so a cancer cell line and also a primary pulmonary cell line. And I'll focus a lot more on the blue cell line here. And then she collected samples at multiple time points post-infection. And what we wanted to see initially is how homogeneous the infection is in these cells, the response of the host. And what we capture at the same time are both the host response to the infection, so in each cell, and also the number of viral transcripts, messenger RNA of viruses that we see in each of these cells. And you can see here in these t-SNE plots, where you have just at 12 hours post-infection, this yellow cluster of cells that has a lot of viral transcripts and a lot that don't. So there's a lot of heterogeneity in the response, even to a high multiplicity of infection. You don't have all the cells that are infected. And at 24 hours, you see most of the cells are infected, but you have a cluster that has very few viral transcripts. Now, when we look at these clusters specifically, what hit us is that actually the cluster where you had the fewer viral transcripts, you had a strong interferon response, which we know is an antiviral response. But we would have expected to see it in a lot of the other cells where you do have viruses. And we see this for the primary cell lines also. But you'll see that certain of these genes that we see don't really correlate with what we would have expected when you do have the strong interferon response. So to get a bit more clarity on exactly what would be the most important genes that are being expressed and controlling other genes, we decided to use a co-expression cluster analysis where we are a network analysis where we would focus on modules of genes. So have these whole groups of genes and how they're related to each other. And we would use the traits that would be the viral, the level of viral transcription. So the number of RNA transcripts that we see corresponding to the viruses specifically. So when we do that and we look at the cluster where we had the fewer viral transcripts, we looked at all those clusters, but one of these clusters is that cluster 2 that had very few viral transcripts. It has a very, like I said, strong response to interferon. And we see that many of the important genes, so the key genes or the hub genes, are interferon stimulated genes. So strong response to interferon, these stimulated genes that we don't necessarily see in the cells that have the actual higher viral transcripts. And we see that in both cell types when we looked. So what is in these clusters of cells that have very few viral transcripts? So when we start looking at the proportion of viral transcripts to defective viral transcripts, we see that there is a higher proportion of these defectives. And here, how we map these defectives was very similar to how we did it before. And what we identified is that overall in the infection, we had a very high abundance of 2 specific DVGs. These are what we named the 162 and 222, and that correlates to basically the position, the coordinate of the first, the gap position where it starts on the segment. And we had done this at high MOI, and we saw there was a high percentage of cells that had a 10 to 20% abundance of these DVGs. And we had also done this experiment at low multiplicity of infection, and we saw that in those cells, there were very few cells infected, but they had mostly a lot of these DVGs, these 2 types of DVGs. So when we go back to our cluster and we look specifically at the cluster that has low viral transcripts, it's basically right here, you have low viral transcripts in this cluster. It actually corresponds to the cluster that has the highest abundance of one of these particular DIs. And so we decided to take these DIs and do reverse genetics. So you can in flu very easily reconstruct the virus, and we put in these DIs specifically in these viruses so that they would have defective polymerases. And then we would culture them with the wild-type virus to see whether they would repress the virus. And that's what we see here. So this is the wild-type virus growing well on its own. And when you co-infect with both of these DIs, you basically get interference of replication of that wild-type virus. So we know these are not just DVGs, they're interfering DVGs, so DIs specifically. So if we go back to our cluster and we look at some of these highly expressed interferon stimulated genes, or ISGs, we see 3 here in particular that we decided to test in culture with these reconstructed viruses that we had. Now, DVGs that you want to culture on their own is difficult because they need a wild-type virus. So we actually use a cell line that expresses the polymerase in trans so that we can have very pure DIs, and we don't need a helper virus. We don't need the wild-type virus. And so we did these experiments where we looked specifically at the expression of ISGs, specific ISGs in a culture where you would infect with pure DIs specifically, and you only look at one round of replication, so they can replicate on their own for one round because there's polymerase that's still around. They just can't go for a second round. So when you do that, you see that your wild type replicates very well at a high level. The DI is not at such a high level, which you would expect. But then when you look at the host response or the cell response to these, you see that there's a low response to the actual virus, wild type virus for these ISGs, but there's a very high response for our DIs. So these DIs are really triggering the ISGs, and it actually has nothing to do with how many of these DIs are there. They have a very strong stimulatory effect. In green here, we use a control that Nigel Dimmock had found years ago. It was this cloned piece that he had found in culture, and basically has done some experiments to look at whether it could be used as an antiviral. And what we observed is that, in fact, the DI we had found in these single cell experiments was much stronger at stimulated the ISGs. Now, induction of ISGs is normally mediated by these type 1 and type 3 interferons, but what we observed here is that they seem to be independent of that. So even in cells where you had a high expression of interferons, you basically, or a low expression of interferons, you would get a high expression of the ISGs. So we're doing experiments going further using knockout mice, and this is just a preliminary where we took a knockout mouse that had a knockout of the one of these receptors, the interferon 1. And what we saw that, in fact, the effect of the DIs, which would be the line on top that looks at the survival post-infection, mice die of these viruses. They die very quickly. And you can see here when they die, this is the wild type virus in both the wild type mice and the knockout mice. But with the DIs or with MOC, which is no virus, you get protection. So the virus do not die, the mice do not die when they're protected with the DIs. So what we're trying to determine in the experiments we're doing is really what is the mechanism of this DI-mediated protection. And we know that it triggers higher ISGs, and the hypothesis was that maybe it triggers it before the virus itself, but it's still an interferon type response. But actually, we think this is more correct, which is actually it bypasses the whole interferon pathway. So the DIs have a completely different way of stimulated the immune response. So I showed you two aspects of, in animals, what do we see in the DIs, and in fact, what is happening at the single cell level in the response to these DIs. So in summary, we do see that these DIs stimulate the response as we saw, but we also see that the host itself, the immune status of the host, can have an effect on these DIs. We also see that the DVGs are not all created equal, and even the DIs are not all created equal. And here I showed you a clean representation where we looked at one specific defective segment, but in fact, multiple segments or different viruses with different segments can be defective. So looking at that effect as a whole can also determine how well or how protective they are. And we see that not cells that have a high DI accumulation or frequency actually respond differently than cells with low DIs. Now, Dimmock has been trying to use in clinical trials now, using DIs as preventive or curative therapy. And for that, what you would need is if I would inject, in your nose, knowing that you're going into an area with high flu circulating, if I could just squirt it in your nose, these population of just very dense DIs, so they're completely defective, no full-length virus, it would have to be pretty stable for a few days to really protect you. And ideally, you would want a therapy that would be transmissible. And so actually, if you can catch a disease, why can't you catch a cure? And that would be a case where maybe STIs that are transmitted would be like a transmitted cure in a population. And that's sort of where I know DMOC and others would like to go, is to be able to transmit these DIs and see whether they're protective. And in fact, DARPA has a very big program, and they call these therapeutic interfering particles. And they're doing this with HIV, multiple viruses, Zika, influenza, and the idea is to be protective. The process is completely different for a retrovirus like HIV, but the idea is sort of interesting. So that's what I wanted to tell you about today. And I mentioned I mentioned Chang and Kate Johnson, another student in my lab who's been doing a lot of the Ferret work. And the single cell work was started by a postdoc in my lab, Bin Zhou, who's now actually heading a flu vaccine group at the CDC. And I have collaborations with the Mount Sinai group, Binjiang, and Christian Forst for some of the network analyses for our single cell data. And the mouse experiments are done in collaboration with Mirella Salvatore at Cornell, and Ted Ross does a lot of these experiments with ferrets at University of Georgia. And this is my group in the respiratory tract. Thank you very much.